We finally know why humans don't have tails. Researchers discover a potential genetic connection between humans' tail loss and the type of birth defect. Approximately 25 million years ago, an ancestor of both humans and apes genetically diverged from monkeys and lost its tail. No one had identified the genetic mutation responsible for this dramatic change in our physiology until now. In a new study published today, February 28, in the journal Nature, researchers identified a unique DNA mutation that drove the loss of our ancestors' tails. It's located in the gene TBXT, which is known to be involved in tail length and in tailed animals. The impressive discovery began when first study author Bo Xia, formerly a graduate student at New York University, who is now a principal investigator at the Broad Institute, injured his tailbone and became interested in the structure's origin. Bo is really a genius because he looked at something that thousands of people at least must have looked at before, but he saw something different, said Itai Yanai, scientific director of the Applied Bioinformatics Laboratory at NYU, Langone Health, a senior author of the study. Jumping genes and dark matter. Over millions of years, changes in DNA allow animals to evolve. Some changes involve only a single rung in DNA's twisted ladder, but others are more complex. The so-called ALU elements are repetitive DNA sequences that can generate bits of RNA, a molecular cousin of DNA, that can convert back to DNA and then insert themselves randomly into the genome. These transposable elements, or jumping genes, can disrupt or enhance the gene's function upon insertion. This specific type of jumping gene exists only in primates and has been driving genetic diversity for millions of years. In this latest study, the researchers found two ALU elements in the gene TBXT that are present in great apes but not in monkeys. These elements are not in the part of the gene that code for proteins, the exons, but rather in introns. Introns are DNA sequence flanking exons that have been referred to as dark matter of the genome because they were historically assumed to have no function. They are removed or spliced out of the sequence before an RNA molecule gets converted into protein. In this case, however, when cells use TBXT gene to generate RNA, the repetitive nature of the ALU sequence causes them to bind together, and this complex structure still gets cut out of the regular RNA molecule, but takes an entire exon with it, thereby changing the final code for and structure of the resulting protein. We did a lot of other analysis of other genes implicating in tail length or morphology, and of course there are differences, but this was like a lightning bolt, said Jeff Boak, director of Institute for System Genetics at NYU Langone Health and senior author of the study. He said, and it was non-coding DNA introns that was 100% conserved in all the apes and 100% absent in all the monkeys, he said. In human cells, the researchers confirmed that the same ALU sequence appear in the TBXT gene and result in removal of the same exon. They also found that the related RNA molecule can be cut in various ways, a variety of ways, to generate multiple proteins from the same gene. By comparison, mice make only one version of the protein, so having both versions seems to prevent the formation of tails, the team concluded. And this way of making different proteins from the same gene is called alternative splicing. It's one of the reasons human physiology is so complex. But this is the first time ALU elements have been shown to cause alternative splicing. Mutations like this have often been thought of limited sequence in evolution, and here the authors show that such a mutation has had a profound impact on our species, said Kirk Lohmiller, professor of ecology and evolutionary biology of human genetics, University of California, Los Angeles. It's exciting to think of how many other complex mutations like this could have generated important traits throughout human evolution, Lohmüller said. Bipedalism and birth defects. The researchers experimented with inserting these same jumping genes into mice, and they found that the mice lost their tails. Notably, evolutionary biologists hypothesized that the loss of the tail allowed humans to become bipedal, according to a 2015 review. We are the only paper that has ever put together a plausible scenario for how it happened, Yanai said. We're now walking on 2 feet, and we evolved a big brain and wheel technology, he said, and from just a selfish element jumping into the intron of a gene. It's astonishing to me, he said. Interestingly, the researchers found that the mice that lost their tails showed a greater prevalence of spina bifida, a birth defect that affects the neural tube, an embryonic structure that gives rise to the spinal cord and brain. The condition affects approximately 1 in 1,000 human births, according to the Centers for Disease Control and Prevention. It may be a sort of unintended consequence that TBXT deficiency gives you a short tail, but it makes it more likely that you don't get that complete neural closure, meaning a hole is left in the neural tube, Bilk said. No one ever thought that, but just following our curiosity, we would make a mouse lose their tail by putting in the same mutation, and then we see the mouse also had a neural tube defect, Yanai said. The discovery of this type of alternative splicing will likely influence the whole field of genetic analysis in the future. I think there's going to be more of them out there, Boak said, of these influential ALU elements. Therefore, he added, there's probably alternatively spliced proteins out there that are actually the root cause of some evolutionary change in our traits. Ever wonder why some people build muscle more easily than others, or why freckles come out in the sun? And this is on Live Science by Jennifer Ziba. Please leave your comments. Thank you for your support. Support my Patreon account. The daily posts are 5 videos daily, and they are totally different from what I have on my YouTube channel. Thank you so much for your support and that you find all my content so interesting. You'll find the Patreon account details in the description box below.